If you were to take a solid steel coin, let's say like a heavy industrial washer, and you just swallow it right now, within just a few hours, the surface of that steel would actually begin to pit and corrode.
Yeah, it really would.
Right, because right at this very second, you were walking around carrying this like fleshy, muscular pouch filled with literally industrial-grade metal melting acid.
It's a terrifying thought when you isolate it like that. I mean, we're housing an environment that is fundamentally hostile to our own tissues.
Right, it's sloshing around just inches below your heart.
Yeah.
And somehow, miraculously, you don't just dissolve from the inside out.
You don't, and there is a brilliant biological reason for that.
Which is exactly why I'm so obsessed with today's deep dive.
Yeah.
Because we walk around completely oblivious to the sheer scale of the engineering happening beneath our skin.
Oh, totally.
We treat our bodies like a rental car, right? Like, oh, it just goes. But what's actually happening in there is a sprawling, nonstop, infinitely complex biological industrial complex.
Yeah.
Exactly. A factory. One that has been continuously refining and perfecting its operations for roughly 200,000 years.
And it's a factory that runs entirely on automation. I mean, there are no holidays, no downtime.
Yeah, and the wild part to me is the management structure of this whole thing. Because you and I, we are the CEOs of this factory. We walk around, we have the title, but we have absolutely zero control over the actual machinery on the factory floor.
Right. You can't
I can't just send a memo down to my pancreas.
Yeah.
Telling it to like pick up the pace today because I ate a heavy lunch.
No, you can't. The autonomic nervous system acts as the ultimate, basically untouchable middle management.
Yeah.
It handles the logistics, the chemical engineering, the waste management, all without ever consulting your conscious brain.
But as the CEO, you do hold one massive absolute power.
You have total executive control over the loading dock.
Yes.
You control the single source of energy this entire complex requires to keep running.
The fuel.
The fuel. What you choose to put into the system, how much of it, and when.
Right.
And understanding the mechanics of what happens once that fuel crosses the loading dock. I mean, that changes everything about how you make those executive decisions.
So that is our mission today. We are going to trace the exact, fascinating, and honestly, mind-bending path of a single ordinary breakfast.
And we have some incredible source material breaking down this entire biological process step by step.
Exactly. We're keeping it classic for this journey. Just a piece of whole wheat toast, drizzled with a little olive oil and topped with a couple slices of ham. Nothing fancy, just a totally normal bite of food.
But the journey that single bite takes is extraordinary. We are going to follow it down a highly specialized 9-meter-long tube, tracking the biochemistry every step of the way.
And the timeline here is the first thing that really trips me up. Because eating that toast takes, what, maybe 10 minutes?
If you're eating slowly, yeah.
Right. But the internal processing of that exact same meal that is going to take your body anywhere from 24 to 72 hours.
It's a massive undertaking.
10 minutes of input for up to 3 days of continuous, highly coordinated industrial breakdown. It's just wild.
It is. But, before we jump into the mouth and start this timeline, we really need to completely reframe how we think about food.
Okay, how so?
Well, because to really grasp what this factory is doing, we have to talk about molecular structures and thermodynamic energy.
Okay, I'm with you, but let's make sure we ground this. Because when we say the word digestion, I think a lot of people just picture food getting crushed up into a smoothie in your stomach.
Yeah, that's a common visual.
But it's much more fundamentally destructive than that, isn't it?
Far more. I mean, your body cannot use a piece of bread. It cannot use a slice of ham.
Right.
It doesn't even recognize them as fuel in that state. Everything you eat is just a vast, complex arrangement of molecules held together by chemical bonds.
So digestion isn't just mashing food.
No, it is the systematic depolymerization of these massive food structures.
Depolymerization, so basically breaking the bonds.
Precisely. Let's look at the proteins in the ham, for example.
Okay.
A protein isn't just a blob of meat. Biochemically, it's a massive, incredibly complex, tangled 3D structure.
Like a knot.
Like an origami sculpture. Imagine a giant, intricate origami sculpture made out of a really long ribbon.
Oh, I like that visual.
Right. And that ribbon is made of amino acids, linked together by strong peptide bonds. Now, your body desperately wants those individual amino acids to build its own structures, but it can't access them while they are folded up into this tight origami shape.
And the bread is a similar structural problem, just a different material.
Right. The bread is whole wheat, which means it's packed with starch,
Ah.
and starch is a polymer. So imagine hundreds of individual glucose molecules, simple sugars, locked together in a tremendously long, rigid chain via what we call glycosidic bonds.
So the ultimate goal of digestion, the whole point of this 9-meter journey, is just getting past the architecture.
Exactly.
It's breaking those peptide bonds in the ham and the glycosidic bonds in the bread. Dismantling these complex structures back down into individual monomers like single amino acids, single sugars that the body can actually absorb and use.
It's tearing down a skyscraper just to salvage the individual steel beams and bricks.
That makes perfect sense. Okay, so with that molecular architecture in mind, let's take that first bite of the toast.
Here we go.
Because there is a huge misconception here that digestion is a stomach-only process. People picture the stomach as this bubbling cauldron where everything happens.
Which is completely wrong.
Right.
The dismantling process begins the absolute second that food crosses the threshold of your mouth.
The front door of the factory.
And the first step is entirely mechanical, driven by your teeth.
Which, by the way, I have to point out for you listening.
Yeah.
This right here, mastication, chewing this, is the only part of this entire 9-meter journey that you actually have voluntary control over.
Enjoy it while it lasts.
Seriously.
Yeah.
But, so why is chewing so important?
Well, that voluntary mechanical step is a prerequisite for all the chemistry that follows. Why? Because of surface area.
Right.
As we just discussed, the goal is to break chemical bonds. But the enzymes, the chemical agents that will actually break those bonds, they can only act on the outside surface of the food.
So if you swallow a chunk of bread, wholly.
The enzymes can only attack the outermost layer. It would take an eternity to break it all down.
Oh, wow. So by chewing, you are exponentially increasing the surface area. You're turning one large chunk into thousands of tiny fragments, exposing all of those internal chemical bonds to the environment.
You are priming the canvas for the chemical wash.
A chemical wash. Okay, I was reading some of the specific research on this from our sources, and the volume of fluid we produce frankly blew my mind.
The saliva.
Yes. Do you know how much saliva a healthy person actually pumps into their mouth in a day?
It is a staggering amount when you measure it out.
It is about a full liter every single day. Picture one of those large 1-liter Nalgene water bottles. You are secreting that entire volume into your mouth from 6 main glands and hundreds of microscopic ones.
And it is not just water acting as a lubricant.
No. Saliva is a highly sophisticated bioactive fluid. It contains mucins to protect the lining of your mouth and throat, but its most critical active ingredients are enzymes.
Let's dive into enzymes, because I know they are biological catalysts, but how are they actually functioning here?
Yeah, that's a good question. Because normally, if I want to break a strong chemical bond in a lab, I have to apply extreme heat or extreme pressure.
Right.
But my mouth is sitting at a mild 98.6 degrees Fahrenheit.
That is the magic of enzymes. In chemistry, every reaction requires a certain amount of activation energy to get started. You're right, usually that means adding heat to force the molecules to react and break apart. If you just left a piece of bread in a glass of water at 98 degrees, the ambient thermal energy would take weeks, maybe months, to break those strong glycosidic bonds in the starch.
Months to digest a cracker. That's crazy.
But an enzyme physically binds to the starch molecule. It grabs it, bends it, and puts mechanical stress on that exact chemical bond.
Oh, I see.
By stressing the bond, the enzyme drastically lowers the activation energy required to break it. Suddenly, the ambient heat of your body is more than enough.
The bond snaps.
The bond snaps. The enzyme lets go, perfectly intact, and moves on to the next bond.
So it's working like a specialized key, unlocking the bond without getting destroyed in the process.
Right, and we have thousands of different types of these enzymes, each highly specific to a certain type of bond.
And your saliva contains a very specific enzyme called salivary amylase.
Yes.
Amylase has one job in the universe.
Yeah.
It looks for the specific glycosidic bonds linking glucose molecules together in a starch chain, and it stresses them until they snap.
So while you are chewing that toast, just mechanically grinding it, the amylase in your saliva is already chemically dismantling the starch architecture.
That's wild. It's snipping the long carbohydrate polymers into smaller and smaller chains of sugar before you even swallow.
It is a simultaneous mechanical and chemical attack. And this isn't just an abstract biochemical concept either. You can actually experience this reaction happening in real time.
Oh, I love this experiment. If you're listening, try this next time you have a piece of plain bread or a cracker.
Yeah, it's very cool.
Take a bite, and instead of chewing and swallowing it in 10 seconds, just keep chewing it. Chew it for a really, really long time, a minute, maybe two, and notice what happens to the flavor.
It suddenly starts to taste distinctly sweet.
Right. That sweetness isn't an illusion.
Yeah.
It is literal biological chemistry happening directly on your tongue.
Because the bread itself isn't sweet, it's starch.
Exactly.
But by chewing it for a minute, you are giving that salivary amylase enough time to completely depolymerize the starch chain. It snaps enough of those bonds that it frees the individual glucose monomers.
And your tongue's taste buds, which couldn't detect the massive starch chain, suddenly register the raw sugar that was locked up inside the bread's architecture all along.
You are tasting the exact moment of chemical digestion.
That is just so cool to me. Okay, so your teeth have crushed the toast, the ham, and the olive oil. The saliva has saturated it. The amylase has started snapping the starch bonds.
Right.
Your tongue gathers this soft, wet, partially digested ball of food, which the biological literature affectionately calls the bolus, and pushes it to the back of your throat.
And this is the point of no return. You trigger the swallowing reflex.
The conscious brain clocks out.
Yep.
The autonomic nervous system takes over the entire operation. We are now entering the involuntary plunge.
The second stop on the journey, the esophagus.
Now, I think the default assumption for most people is that the esophagus is essentially just a biological drain pipe.
Oh, absolutely.
You swallow, gravity takes over, and the food just sort of falls down into your stomach, like dropping a rock down a well.
That is a very common misconception, but from a biomechanical standpoint, it is entirely false. The esophagus is not a passive pipe.
It's not.
No, it is a highly active muscular tube, about 25 centimeters long, and it relies absolutely 0% on gravity.
The analogy that makes this click for me is a fresh tube of toothpaste.
Ah, yes.
If you take the cap off a tube of toothpaste and hold it upside down, the paste doesn't just fall out. Gravity isn't enough. You have to actively squeeze the tube from the top and continuously run your fingers down the plastic to force the paste forward.
That is exactly what the smooth muscle tissue of your esophagus is doing to the bolus.
Wow.
The moment the food enters the top of the tube, the muscles just above the food contract tightly. Then that ring of contraction moves downward in a synchronized rhythmic wave. This is called peristalsis.
Wait, so this peristaltic wave is actively crushing the food downward. Does that mean, theoretically, I could eat while hanging entirely upside down?
Not just theoretically. Physiologically, you could hang by your knees from a set of monkey bars, take a bite of your toast, chew it, swallow it, and the peristaltic wave of your esophagus would effortlessly squeeze that bolus up your chest, working completely against gravity, and push it straight into your stomach.
I mean, I don't recommend trying that for breakfast, but it perfectly highlights the sheer mechanical force we are dealing with.
It is an active transport system.
And it's fast. It's an 8-second transit time through that 25-centimeter tube.
But at the very bottom, right before the bolus drops into the stomach, it hits a critical security checkpoint.
The lower esophageal sphincter.
Yes. It's a specialized ring of muscle that acts as a tight one-way security valve.
Right.
Under normal conditions, it senses the approaching peristaltic wave, relaxes for just a fraction of a second to let the bolus drop into the stomach, and then immediately snaps shut again with intense pressure.
And this is where the system can fail for millions of people, right? Because when we talk about heartburn or gastroesophageal reflux disease, GERD, people often frame it as a chemical problem.
Like they have too much acid.
Exactly. Oh, my stomach is just producing way too much acid. But looking at the mechanics of this, that's not the core issue at all.
Usually no. Acid reflux is fundamentally a mechanical failure, not a chemical overproduction.
Okay, walk us through that.
That lower esophageal sphincter, that security valve, weakens over time, or it spasms and relaxes when it shouldn't.
So the stomach acid isn't increasing in volume, it's just escaping its designated containment zone.
Exactly. The stomach is built to handle the acid. The esophagus is not.
It lacks the specialized protective lining.
Exactly. So when that valve fails, even a tiny splash of normal stomach acid reaching up into the lower esophagus causes severe chemical burns to the tissue. That is the pain of heartburn.
Which brings us ominously to the stomach itself, the biological concrete mixer.
The stomach is a marvel of evolutionary engineering. It's essentially a J-shaped, heavily muscled bag sitting slightly to the left of your upper abdomen.
And it is incredibly elastic. When it's empty, it's crumpled up and only about the size of your closed fist.
Very small.
But as you eat that breakfast, the folds of the rugae smooth out, allowing the stomach to expand to comfortably hold over a liter, sometimes up to a liter and a half, of food and liquid.
But the volume isn't what makes the stomach truly unique. What makes it unique is the environment it generates inside that bag.
The hostile zone.
Exactly. The moment your toast drops in, it is entering a deeply hostile, highly volatile zone. The parietal cells lining the stomach immediately begin pumping out hydrochloric acid.
Let's pause here and really contextualize how strong this hydrochloric acid actually is, because we hear the word acid all the time, and the pH scale can feel a bit abstract to non-chemists.
Right. The pH scale measures the concentration of hydrogen ions in a liquid, essentially how acidic or alkaline it is. It runs from 0 to 14. Pure water sits right in the dead center at a neutral 7.
And the scale is logarithmic, right? Meaning a pH of 6 isn't just a little more acidic than 7, it's 10 times more acidic. A pH of 5 is 100 times more acidic.
Exactly.
So the lower the number, the more fiercely corrosive it is. Lemon juice, which is pretty sharp, sits around a pH of 2. Where does our internal stomach acid land?
When actively digesting, human stomach acid typically ranges between a pH of 1.5 and 3.5.
It is exponentially stronger than lemon juice.
It is.
We are talking about a fluid that is literally strong enough to pit and corrode a piece of solid steel. It is the exact same chemical formulation, hydrochloric acid, that industrial laboratories use to dissolve raw metals and clean masonry.
Yeah, the exact same stuff.
And we are just casually walking around with a pouch of it sloshing beneath our ribs. How does this not just melt right through our internal organs?
It absolutely would, and very quickly if it weren't for a highly specialized continuous defense mechanism.
The mucus.
Yes. The internal epithelial cells of your stomach are constantly secreting a very thick, dense layer of highly alkaline mucus.
Alkaline, meaning it's on the opposite end of the pH scale.
Yes. This mucus is rich in bicarbonate ions. It creates a physical and chemical barrier roughly a millimeter thick across the entire inner surface of the stomach.
Okay, so it's a shield.
Exactly. So as the steel-melting hydrochloric acid approaches the stomach wall, it hits this alkaline mucus, and an immediate neutralization reaction occurs at the boundary. The acid literally cannot penetrate to the tissue.
The stomach has to constantly, continuously defend itself against its own internal weaponry.
And the moment it stops producing that mucus,
You get an ulcer.
the acid just burns a hole right into you.
That is wild, but it begs a massive evolutionary question for me. Why do we need steel-melting acid in our bodies in the first place? Is it just to dissolve the toast?
Dissolving the food is only a secondary benefit. The extreme acidity serves two primary indispensable functions.
Okay.
The first function is actually immunological. It's a frontline defense system.
A biological incinerator.
Exactly. Every single time you eat, no matter how clean your kitchen is, you are ingesting millions of bacteria, viruses, and environmental pathogens.
Oh, true.
If those microbes reached your intestines alive, you would be in constant peril of severe infection. The extreme hydrochloric acid bath instantly sterilizes your food.
Oh, wow.
The low pH destroys the cell walls of the vast majority of those microscopic invaders before they can ever take hold.
Well, just sanitizing the toast.
Yes.
But you said there was a second function, which brings us back to our molecular architecture.
Yes, chemical preparation. We talked about salivary amylase cutting the starch bonds in the mouth, but amylase does absolutely nothing to the ham on your toast.
Right. Ham is dense animal protein.
Our complex 3D origami sculpture is made of amino acids.
Right, right.
And this is where the physics of the acid comes into play. When that complex, tightly folded protein origami drops into a pH of 1.5, the intense acidic environment disrupts all the delicate hydrogen bonds holding the 3D shape together.
So it falls apart.
The protein undergoes denaturation.
Denaturation. So the origami sculpture is violently forced to unfold. It flattens out into a long, single, continuous ribbon.
Exactly. The protein loses its 3D shape and unravels into a straight polypeptide chain. And this unraveling is absolutely crucial because only when the ribbon is laid out flat are the strong peptide bonds linking the individual amino acids exposed.
Ah. So the acid isn't actually cutting the protein apart, it's just untangling it so the actual scissors can get to work.
Precisely. And the stomach provides its own highly specialized pair of chemical scissors.
Enter the enzymes.
The stomach lining secretes an inactive substance called pepsinogen. Now, pepsinogen is completely harmless on its own, but the moment it encounters the extreme hydrochloric acid, the acid chemically cleaves a piece off the pepsinogen molecule, transforming it into its active, aggressive form, pepsin,
And pepsin is an enzyme designed specifically for one job.
to attack peptide bonds. Once the acid has denatured and unfolded the protein origami into a flat ribbon, the pepsin swoops in and starts aggressively snapping the peptide bonds,
Snipping it all up.
chopping that massive ribbon into much shorter, more manageable fragments called peptides.
It doesn't break them down into the single individual amino acids just yet, but it chops the massive structure into bite-sized chains.
Right?
And all while this highly precise biochemistry is happening, the stomach is putting on a violent physical show.
It is a mechanical tempest in there.
The muscular walls of the stomach are structured in 3 different layers: longitudinal, circular, and oblique, allowing it to contract and twist in multiple directions simultaneously.
It's churning, squeezing, and mashing the chewed toast, the ham, the olive oil, the acid, and the pepsin together.
And this intense, aggressive churning phase lasts anywhere from 2 to 4 straight hours.
It's a brutal environment designed for maximum disruption.
And the result of this 2 to 4 hour acid assault is that your breakfast is completely unrecognizable. The toast, the oil, the ham has all been homogenized into a thick, highly acidic, grayish, semi-liquid paste called chyme.
Chyme is the perfect biological term for it. But here's something that often surprises people. Despite all this extreme mechanical and chemical action, your stomach actually absorbs almost nothing into your bloodstream.
Wait, really? We've spent 4 hours breaking this down and we aren't getting any energy from it yet.
With very few exceptions, alcohol being a notable one, which can pass through the stomach lining, and certain medications like aspirin, the stomach is not an organ of absorption.
No nutrients.
No nutrients, no amino acids, no glucose enter your body here. The stomach is strictly a decontamination chamber and a prep station.
It's purely preparing the chyme for the next stage. And it executes the handoff really methodically.
It does.
At the very bottom of the J-shaped bag is another muscular valve, the pyloric sphincter. Now, the stomach doesn't just open the floodgates and dump a liter of this highly acidic, churning chyme into the next organ all at once. That would be catastrophic.
It would overwhelm the system entirely.
Right.
Instead, the pyloric sphincter acts as a meticulous metering valve. It squirts the chyme out incredibly slowly, roughly 3 milliliters, about half a teaspoon at a time.
And the pacing of this release is fascinating because it's actively dictated by the chemical composition of the meal you just ate.
The neurohormonal feedback loops here are incredibly sensitive.
So if you just drink a large glass of water on an empty stomach, the stomach says, great, nothing to denature here, and it flushes it through the pylorus into the intestines in a matter of minutes.
Right.
But our breakfast has olive oil and ham, fats and proteins.
And fats and proteins are complex molecules that require far more time to emulsify and denature. As the chyme enters the first part of the intestine, sensors detect the high concentration of fats and proteins and send hormonal signals like cholecystokinin back to the stomach,
Telling it to slow down.
telling the pyloric sphincter to tighten up and telling the stomach muscles to dial back the contractions. It actively delays gastric emptying.
See, I love understanding the biology behind everyday feelings. Because when you go out and eat a massive, rich meal like a heavy steak or a ton of fried food, and an hour later you groan and say, "Ooh, that is really sitting heavy in my stomach," you aren't just using a metaphor.
No, not at all.
It is a literal physical reality.
Uh-huh.
Your body has biochemically locked the exit valve. Your stomach is physically distended, holding on to that heavy load to ensure the proteins and fats are adequately prepped before it safely releases them.
It is the ultimate bottleneck for quality control. Which brings us, finally, to the moment that pyloric valve opens and that half teaspoon of acidic chyme enters the next phase of the journey,
The main event.
a main event.
We are leaving the attack zone and we are entering the laboratory of extraction, the small intestine.
Everything we have discussed up to this point, the chewing, the saliva, the peristalsis, the acid, the pepsin, was purely preparatory. The small intestine is where the actual miracle of biology occurs. This is a highly specialized tube, roughly 6 meters long, about 20 feet coiled up tightly inside the center of your abdomen.
And I have to take a quick detour here because we need to bust a very persistent biological myth from the sources.
Oh, the tennis court myth.
Yes. If you, the listener, took a high school biology class a decade or two ago, you were probably taught a very specific, mind-blowing, fun fact about the small intestine. You learned that the internal wall isn't a smooth pipe. It's covered in deep circular folds.
Right.
And those folds are covered in millions of little finger-like projections called villi. And the cells of those villi are covered in even smaller microscopic hairs called microvilli.
An exquisite fractal architecture, all evolutionarily designed to maximize the internal surface area for the absorption of nutrients.
Right. And the old textbooks used to confidently state that if you took all those microscopic folds, villi and microvilli, and ironed them out completely flat, the total surface area of your small intestine would be the size of a doubles tennis court. Something like 200 to 400 square meters.
It's a fantastic visual. It's been repeated in documentaries for decades. The only problem is it's scientifically outdated.
Completely busted. I was looking at the revised measurements published in 2014 by a team of Scandinavian researchers, Hylander and Fandrix.
Yeah.
They realized the old estimates were based on flawed extrapolations. They used much more precise microscopic imaging and tissue analysis, and it turns out that true surface area isn't a tennis court at all. It's about 30 square meters.
Which, to be fair, is still a massive amount of surface area to pack into a human abdomen.
Oh, absolutely. 30 square meters is roughly the size of a small studio apartment or a two-car garage. You're carrying a studio apartment's worth of highly absorptive tissue inside your belly.
That's still incredible.
It's just not Wimbledon. But here is the immediate crisis. When that half teaspoon of chyme squirts out of the stomach and enters the first section of the small intestine, the duodenum, we have a massive biological problem.
A critical emergency, really.
The chyme is still saturated with hydrochloric acid. It is sitting at a pH of roughly 2.
Highly acidic.
But the small intestine does not possess that thick, specialized alkaline mucus layer that the stomach has.
No, it doesn't.
If that battery acid is left unchecked in the duodenum for even a few minutes, it will begin to violently digest the delicate microvilli. It will burn straight through the intestinal wall.
The environment must be neutralized immediately. We need a chemical fire extinguisher.
And that is exactly what the pancreas provides. The pancreas is an accessory organ tucked quietly right behind the lower part of the stomach. The absolute split second that acidic chyme breaches the pyloric valve, the pancreas dumps a massive concentrated dose of alkaline bicarbonate fluid into the duodenum.
Bicarbonate, essentially a highly potent biological baking soda.
The chemistry is instantaneous. The alkaline bicarbonate hits the hydrochloric acid and they neutralize each other.
Just like a volcano science experiment.
Exactly. The pH rapidly shoots up from a deadly 2 back to a safe neutral 7. The acid threat is completely neutralized.
Crisis averted. But the pancreas isn't just the fire department. It is also the heavy artillery for the actual digestion process.
Okay, lay it on us.
Because once the environment is safe, the pancreas follows up the bicarbonate by dropping a brand new, highly specialized cocktail of chemical scissors into the mix.
This is where the definitive chemical breakdown happens. The saliva in the stomach just soften the targets. Now the pancreas releases its arsenal.
Yes!
It releases a whole suite of powerful proteases like trypsin and chymotrypsin. These pick up where the stomach's pepsin left off, ruthlessly snipping the remaining peptide chains into individual single amino acid monomers.
It also releases a fresh wave of pancreatic amylase. If any starch chains manage to survive the mouth and the stomach, this amylase hunts them down and snaps the final glycosidic bonds, reducing everything to single glucose molecules.
And crucially, the pancreas releases lipases,
The chemical scissors specifically designed to attack and dismantle fat molecules.
the olive oil from our toast. But wait, I have a big pushback here, just looking at the fundamental laws of physics.
Oh, really? Okay.
We've established that the inside of the small intestine is essentially a watery fluid environment, right?
Yeah.
It's all liquid chyme and water-based pancreatic juices.
Correct.
Well, anyone who has ever tried to make a vinaigrette salad dressing knows the fundamental rule of the universe. Oil and water do not mix.
Very true.
The oil is hydrophobic. It just clumps together, separating from the water into these big floating globs. So how on earth do these watery, pancreatic lipase scissors get a grip on the globs of olive oil to cut the chemical bonds? Wouldn't the enzymes just slide right off the surface of the oil glob?
They absolutely would. The lipases are water-soluble, meaning they can only operate at the interface where the water meets the oil.
Right.
If the fat is clumped together into one massive glob, the surface area is incredibly small compared to the volume. The lipases would be largely powerless. They would just bounce off. The body has to somehow solve the oil and water thermodynamics problem.
And it solves it brilliantly by calling in another helper organ, the liver, acting through its specialized storage pouch, the gallbladder.
The liver sends in the necessary reinforcements to change the physical state of the fat.
Yes. The liver continuously manufactures a thick, yellowish-green fluid called bile. It stores this bile in the gallbladder. When the fat from the olive oil enters the intestine, The gallbladder contracts and squirts this bile directly into the chyme.
And the bile salts act exactly, and I mean chemically, almost identically to dish soap.
It is the exact same chemical principle. Amphipathic molecules. Let's explain that because this is so cool.
Go for it.
Think about when you have a frying pan coated in thick, stubborn bacon grease, and you run water over it. The water just beads up and rolls off. The grease stays perfectly intact.
Right.
But the second you add a single drop of dish soap to the pan,
The grease instantly shatters. It rapidly breaks apart into a cloudy suspension of millions of tiny droplets.
emulsification.
Emulsification. The bile salts, just like the dish soap, are amphipathic. This means they have a split personality at the molecular level.
Oh, I see.
One end of the bile salt molecule is hydrophilic. It deeply loves water. The other end is hydrophobic. It loves fat and hates water.
So when the bile hits the big glob of olive oil, the fat-loving ends wedge themselves deep into the oil, while the water-loving ends face outward toward the watery chyme.
The physical tension literally tears the massive glob of oil apart into microscopic nanometer-sized spheres called micelles.
It vastly exponentially increases the surface area of the oil.
And suddenly, the oil and water problem is solved. The pancreatic lipases, the fat scissors, now have infinite access.
Well, that's brilliant.
They swarm these millions of microscopic micelles attached to the surface and rapidly dismantle the complex fat molecules, the triglycerides, into simple fatty acids and glycerol.
So let's pull back and look at the big picture of what is happening in the small intestine right now. It is a masterpiece of synchronized, multi-front biochemical warfare.
It is total molecular deconstruction. The architecture of the food has been entirely defeated.
Let's track the final molecular fate of our breakfast. Number 1, the toast. The complex starch polymers have finally been completely depolymerized. There are now millions of individual glucose molecules floating freely in the fluid. Quick, accessible, pure cellular fuel.
Number 2, the ham. The tangled, complex origami protein sculptures, having been denatured by acid and chopped by pepsin, have now been completely dismantled by the pancreatic proteases into individual amino acids, ready to be used as raw building materials for muscle repair, enzymes, or hormones.
And number 3, the olive oil. Shattered into micelles by the bile salts, the triglycerides have been snipped by lipases into free fatty acids. Dense, highly packable energy storage.
The deconstruction is complete. The towering skyscraper has been reduced to neat, sorted piles of individual steel beams, bolts, and glass panes.
And this leads to what I think is the most philosophically profound moment in the entire biological process. The moment of true transformation.
I completely agree.
Because once these molecules are reduced to their fundamental monomer state, single sugars, single amino acids, single fatty acids, they are finally small enough to pass right through the cellular walls of the small intestine.
They are actively transported across the membranes of the microvilli, absorbed across those 30 square meters of surface area.
They just pass right through.
The glucose and amino acids slip directly into the capillaries, entering the bloodstream. The fatty acids are repackaged and slip into the lymphatic system, which will eventually drain into the blood.
I just want you listening to this to really sit with the weight of this realization for a second. Think about the physical boundary of your body. 3 or 4 hours ago, that was just a piece of toast, some pressed olives, and some ham sitting on a ceramic plate.
Yeah.
It was a foreign object. It was part of the outside world. And now it has crossed the ultimate threshold. It is glucose speeding through your veins, crossing the blood-brain barrier to fuel the neurons in your cortex so you can comprehend this sentence I am speaking right now.
That's amazing.
It is amino acids being rushed to your bicep to physically rebuild the micro tears from the heavy box you lifted yesterday. It is fatty acids being tucked away in your adipose tissue to ensure you survive if you don't eat tomorrow.
The barrier between the outside world and you has entirely dissolved. The food hasn't just fueled you. The physical atoms that made up that bread and that meat have literally, materially become you.
It is biological alchemy. The sheer intimacy of eating is wild when you look at it molecularly.
It really is.
But okay, let's keep moving. Because if the small intestine extracts all the good stuff, all the usable glucose, amino acids, and fats, what happens to the chyme that is left over?
The leftovers.
The fluid keeps moving down the tube into the next section. Are we just dealing with biological garbage at this point?
Calling it garbage would be a massive insult to the evolutionary brilliance of the next 2 stages of the process. The leftovers are absolutely critical to your survival.
Really?
Yes. As the remaining unabsorbed slurry pushes through the final valve of the small intestine, the ileocecal valve, it enters the large intestine, commonly known as the colon.
The cleanup crew.
It's a much wider, thicker tube, about 1.5 meters long, framing your abdomen, and it has two desperate, utterly vital jobs. The first is a matter of sheer fluid mechanics, water recovery.
Right, because we have to remember how much liquid the factory pumped into this tube to make all this chemistry happen.
A lot.
We had the liter of saliva, we had a liter or two of gastric acid, we had the bile from the liver, we had the bicarbonate and juices from the pancreas.
Yeah.
The biological accounting notes that your digestive system pumps anywhere from 7 to 9 liters of internal fluid into the GI tract every single day just to move the food along and facilitate these chemical reactions.
It is an immense volume of water being drawn from your blood plasma, and if your body just let all of that fluid exit the system as waste at the end of the line,
You would die.
you would be dead from severe, catastrophic hypovolemic shock and dehydration in a matter of hours. The factory would run completely dry. Your blood pressure would plummet to 0.
Wow.
So the large intestine acts as a massive, highly efficient osmotic sponge. As the liquid leftovers slowly move through this 1.5-meter tube over the course of 12 to 36 hours, the cells lining the colon furiously pump sodium ions across the membrane.
And the water following.
And the water follows via osmosis, sucking the fluid back into the bloodstream to be recycled and used again.
It is the ultimate closed-loop recycling system. But water recovery is only the first job. The second job of the large intestine involves what is arguably the most complex, mysterious, and powerful workforce in the entire factory.
The microbiome, the internal ecosystem.
This breaks my brain every time I think about it. Your large intestine is not just a hollow tube. It is a thriving, densely populated alien ecosystem.
Yep.
Trillions of microscopic organisms live in there.
Trillions.
In fact, if you count them up, there are literally more bacterial cells living inside your large intestine than there are human cells in your entire body. From a purely numerical standpoint, you are more them than you are you.
And they are not just hitchhikers. They aren't parasites taking advantage of a warm, wet environment. They have a symbiotic, deeply integrated, millions of years old relationship with your factory. They eat what you are biologically incapable of digesting.
Okay, what does that mean in the context of our breakfast? Yeah. We had the enzymes to break down the starch, the proteases for the protein, the lipases for the fat. What could possibly be left that we can't digest?
The structural architecture of the plant. You ate whole wheat toast.
Right.
The whole wheat grain is surrounded by a tough, complex carbohydrate matrix called cellulose, or dietary fiber. Human beings, despite all our sophisticated evolutionary engineering, do not possess a single microscopic scissor. Not one enzyme capable of breaking the specific beta-glycosidic bonds that hold plant fiber together.
It's a molecular puzzle we just don't have the key for.
Exactly. So that fiber passes through the crushing of the teeth. It survives the steel-melting acid of the stomach. It ignores the pancreatic enzymes in the small intestine, and it arrives in the large intestine completely intact.
But when it hits the colon, the microbial workforce is waiting.
Because they have the keys.
They possess the vast genetic library of enzymes required to dismantle cellulose.
They feast on the fiber. And because there is very little oxygen deep inside the large intestine, they break down this fiber through an anaerobic process called fermentation.
And this isn't just about them getting a free meal. The byproduct of this bacterial fermentation is actually crucial for our health. The bacteria are essentially operating a secondary chemical plant inside us, producing entirely new molecules from the fiber.
Yes. They manufacture a specific family of molecules called short-chain fatty acids, or SCFAs. Acetate, propionate, and specifically butyrate.
These bacteria literally manufacture high-grade cellular fuel out of the indigestible trash.
They do. Butyrate, for example, is the primary preferred energy source for the human cells that line your colon.
Oh, wow.
The bacteria are literally paying rent by feeding the cells that house them, keeping the intestinal barrier strong, and preventing leaky gut.
That's a great deal for us.
But beyond local maintenance, these short-chain fatty acids are absorbed into your main bloodstream, where they act as incredibly powerful epigenetic modulators and signaling molecules.
Like the bacteria are sending chemical text messages to the rest of the human body.
They absolutely are. They interact directly with your immune system, modulating T cells and actively suppressing systemic inflammation.
And the brain, too, right?
Yes. And science is just now mapping the profound implications of the gut-brain axis. These bacterial byproducts can cross the blood-brain barrier. They interact with the vagus nerve. They influence the production of neurotransmitters like serotonin.
It is insane.
They impact how your body stores fat, and they can actively alter your mood, your anxiety levels, and your cognitive function.
So to draw a straight line here, the alien bacteria in your gut are eating the indigestible fiber from the wheat on your toast, and in return, they are synthesizing chemicals that circulate to your brain and might literally make you feel happier, less anxious, or more focused.
Yep.
The leftovers are actively influencing the CEO in the control room.
It completely reframes the concept of biological waste. What is finally left over at the very end of the line after the water is wrung out and the bacteria have... To understand the primacy of the liver, you have to look at the internal plumbing of the human body. We talked about all those extracted monomers, like the thousands of glucose molecules, the amino acids being absorbed through the walls of the small intestine directly into the bloodstream.
Right.
But that heavily enriched, nutrient-dense blood does not just enter the general circulation, get pumped to your heart, and send out to the body indiscriminately. That would be metabolic chaos.
Well, it would be like a supplier dumping millions of raw, unsorted parts directly onto an active factory floor without a warehouse manager telling anyone where they go or if they're even needed.
Exactly. The body cannot tolerate that kind of biochemical volatility. So instead, evolutionary plumbing has routed all of the veins carrying blood away from the intestines to converge into one massive, single vascular superhighway, the hepatic portal vein.
Okay.
And that portal vein leads directly and exclusively to the liver.
The liver gets absolute first dibs on everything you absorb.
It is the ultimate security filter, the chemical processing plant, and the warehouse manager all rolled into one. It's a massive, dark red, 1.5 kilogram organ sitting under your right ribs, and it performs over 500 distinct vital biochemical functions.
500.
It actively decides the fate of every single molecule that comes from your toast.
Okay, let's walk through the liver's specific decision-making process for our breakfast, molecule by molecule. Let's start with the bread.
Okay.
The complex starch has been depolymerized into thousands of individual glucose molecules. The liver sees this massive, sudden spike of pure sugar arriving via the portal vein. What does it do with it?
The liver immediately acts as a metabolic dam. If it let all that raw glucose flow straight into your main bloodstream, your blood sugar levels would skyrocket to acutely toxic, lethal levels. The osmotic pressure would destroy your blood vessels.
So it blocks it.
So the liver stops the vast majority of it. It allows only a steady, highly regulated trickle of glucose to pass through into general circulation, just enough to keep your brain firing and your immediate energy needs met.
But it has to put the rest of that sugar somewhere. It can't just delete the molecules.
It stores them, and this is a beautiful piece of chemistry. The liver takes the individual glucose monomers and begins linking them back together.
Rebuilding them.
Yes. It synthesizes a brand new, highly branched polymer called glycogen. It packs this glycogen tightly inside its own cellular vaults. Glycogen is essentially a biological battery, a quick-release energy reserve.
Like a supercapacitor.
Exactly. If you are sitting on the couch now, but you suddenly need to sprint for a bus an hour from now, the liver detects the adrenaline, instantly snaps the glycogen bonds back into glucose, and floods your blood with the exact amount of energy required for the sprint.
It's a perfect system. But here is the critical evolutionary constraint, and I think anyone who has ever struggled with their weight needs to hear this. The warehouse is not infinite.
Far from it. It is actually shockingly small. The human liver can only store roughly 100 grams of glycogen at maximum capacity.
That's it.
Your skeletal muscles can store maybe another 400 grams or so locally. That's it. Roughly 500 grams of quick sugar storage for the entire human body.
Which made perfect sense for 199,000 years of human evolution when finding a handful of sweet berries was a rare miracle. But today, you can drink 100 grams of liquid sugar in a single 32-ounce soda.
Very easily.
So what happens biochemically when you eat a massive meal or drink 3 sodas and those limited glycogen warehouses are completely full? The liver can't just throw the extra sugar in the trash.
It doesn't. And this right here is where the fundamental biology of weight gain actually happens. If the glycogen vaults are topped off, but sugar keeps pouring in from the portal vein, the liver initiates a different, highly complex metabolic pathway called de novo lipogenesis.
De novo lipogenesis.
Literally making new fat from scratch. The liver chemically deconstructs the excess glucose molecules, rearranges their carbon, hydrogen, and oxygen atoms, and stitches them together to synthesize brand new fatty acids.
Boom. Excess sugar is literally transformed into fat, period. Once the short-term warehouse is full, The factory just starts manufacturing long-term storage crates. It doesn't matter that you didn't eat fat, you ate sugar, and the liver turned it into fat.
Exactly. It packages these newly minted fats into transport proteins and ships them off to your adipose tissue, your belly, your thighs, for indefinite, unlimited storage.
Okay, now what about the ham? The amino acids from the protein.
Right, the liver's triage of the protein beads.
Yes.
The liver looks at the incoming flood of amino acids and essentially reads the body's physiological work orders. It checks the systemic demand. Does the bicep need repair from exercise? Send a batch of amino acids there.
Makes sense.
Do we need to manufacture more immune cells? Send them to the bone marrow. Are we low on specific enzymes? Keep some here to build them. It routes the structural building blocks exactly where they are actively needed.
But what if there aren't any active work orders? What if you bought a giant tub of protein powder and you drank 100 grams of protein, but you haven't worked out in a week, and your body doesn't actually need to build any new tissue?
The body does not store extra protein for later use. There is no protein warehouse.
Oh, really?
The liver is ruthlessly efficient. It will not let that potential thermodynamic energy go to waste. Through a process called deamination, the liver strips the nitrogen component off the excess amino acids. That nitrogen is toxic, so the liver converts it into urea, which is sent to the kidneys, and you eventually pee it out.
And what about the rest of the molecule once the nitrogen is gone?
The remaining carbon skeleton of the amino acid is then converted either directly into glucose for immediate energy, or if the energy isn't needed, it is routed right back into de novo lipogenesis and converted into fat for storage.
It all comes back to energy management. Extra protein doesn't automatically become extra muscle. If there's no demand, extra protein just becomes expensive urine and extra fat.
Exactly.
And speaking of fat, what about the actual olive oil we absorbed? The fatty acids that came through the lymphatic system?
The liver pulls those circulating fats in, repackages them into highly specialized transport vehicles called lipoproteins like VLDL and LDL, and sends them back out into the blood to be delivered safely to fat cells for storage. Or to organs like the heart that actually prefer burning fat for dense, sustained energy.
Right.
But there is a fascinating, heavily misunderstood twist about the liver's relationship with lipids.
Oh, this is the cholesterol paradigm shift from the sources.
Yes. When the average person hears the word cholesterol, they immediately associate it with the villain of health. They picture clogged arteries, heart attacks, and greasy food.
Sure.
But from a biological standpoint, cholesterol is an absolute fundamental necessity for human life. It provides the structural integrity and fluidity of every single cell membrane in your entire body.
Wow.
Furthermore, it is the base precursor molecule required to synthesize vitamin D, cortisol, testosterone, and estrogen. You literally cannot exist without a constant supply of cholesterol.
And the really surprising mechanistic fact here is that the cholesterol you physically eat, like the cholesterol in an egg yolk or a piece of shrimp, has surprisingly little physiological impact on your actual blood cholesterol levels.
Because the liver is running the show, and it desires homeostasis above all else. Your liver synthesizes the vast majority of the cholesterol your body uses, roughly 80% of it from scratch, every single day.
80%.
If you eat a meal very high in dietary cholesterol, the liver's sensors detect it, and it simply dials down its own internal production to keep the overall systemic levels balanced.
Ah, so it autoregulates.
Exactly. If you eat a strict vegan diet with zero dietary cholesterol, the liver simply ramps up its own production to ensure the body has what it needs. It is a tightly regulated internal thermostat.
The liver is just sitting there in the dark, pulling all the metabolic levers, keeping the whole ship afloat.
It's the mastermind.
But it has a vulnerability, because it isn't just a warehouse and a synthesizer. It's the security filter. It is responsible for neutralizing toxins that cross the barrier. And there is one widely consumed toxin that perfectly illustrates how fragile this balance is.
Alcohol.
Alcohol.
It is arguably the most common toxin human beings ingest voluntarily, and its metabolic priority overrides almost everything else.
When you drink a beer or a glass of wine with your dinner, the alcohol ethanol is an incredibly tiny, simple molecule. It doesn't need to be digested.
No, it just passes right through.
It is absorbed rapidly, some of it directly through the stomach lining, the rest instantly in the duodenum. And it hits the portal vein and arrives at the liver almost immediately.
And the liver's chemical sensors recognize ethanol for exactly what it is, a highly reactive metabolic poison, a priority 1 cellular threat that can damage tissues and DNA if left circulating.
And this is the stark mechanistic takeaway for you listening.
Hmm.
When the liver encounters alcohol, it sounds the biological alarm and halts almost every other metabolic operation on the factory floor.
The liver has a limited processing capacity. It possesses specific enzymes, alcohol dehydrogenase, to break down the ethanol into less toxic substances and eventually into water and carbon dioxide.
Right.
But to do this, it drops everything else. It prioritizes the dismantling of the poison above all routine maintenance.
Which means, and this is a huge realization for anyone trying to manage their body composition, while the liver is busy fighting the alcohol fire, it completely stops burning fat. It hits pause on your baseline metabolism.
The biochemical pathways required for fat oxidation, beta oxidation, are quite literally chemically blocked by the presence of alcohol metabolism. The liver cannot multitask those two processes efficiently.
So when we talk about alcohol impacting your weight, it isn't just about the empty calories in the beer or the wine itself. It is the cascading systemic effect.
Yeah.
You have forced the CEO of your metabolism to clock out and deal with the toxic crisis.
Yeah.
It means all the incoming fat and sugar from the meal you were eating alongside that drink has nowhere to go.
The liver can't process it for energy, so it defaults to the easiest, safest option. It routes it directly into fat storage.
It perfectly highlights just how profoundly interconnected every single molecular choice is in this system. You cannot isolate one variable without affecting the entire factory floor.
It really does. Everything is connected.
So let's pull all the way back. Let's look at the grand recap of what we've just explored, the sheer scale of this biological automation.
You sit down at a table. You eat a piece of toast with a drizzle of olive oil and a slice of ham. It occupies 10 minutes of your day.
And you trigger a monumental, unstoppable cascade. You initiate mechanical crushing and the precise enzymatic catalysis of starch in the mouth. You launch an 8-second, gravity-defying peristaltic wave down the muscular tube of the esophagus.
You subject the food to 2 to 4 hours of violent mechanical churning and steel-melting hydrochloric acid assault in the stomach, denaturing the 3D protein architecture so pepsin can chop it apart.
You demand 4 to 6 hours of delicate, synchronized extraction in the 30 square meters of the small intestine, neutralizing acid with pancreatic bicarbonate, shattering oil globs with amphipathic bile salts, and absorbing the fundamental monomers across the cellular boundary into your blood.
All while the liver acts as a tireless, brilliant warehouse manager, filtering poisons, synthesizing glycogen, manufacturing fat, and routing amino acids based on systemic real-time demand.
And finally, you rely on 12 to 36 hours of desperate osmotic water recovery and anaerobic bacterial fermentation in the dark, alien ecosystem of the large intestine, allowing trillions of microbes to feed your cells and modulate your brain chemistry.
All of this running flawlessly, silently. Whether you are deeply focused on a complex spreadsheet, sleeping soundly in your bed, or having a stressful argument with a friend,
It's just happening.
your autonomic nervous system and your organs are executing a masterpiece of thermodynamic and industrial logistics multiple times a day, every single day of your life.
It is awe-inspiring. Biological molecular magic. But as we wrap up this deep dive, I want to leave you with one final lingering thought. A conceptual mystery that our discussion today points toward, but doesn't quite fully resolve.
The ultimate philosophical question of the fuel.
Exactly. Because we just spent this entire time explaining exactly how the body physically dismantles that piece of toast into single molecules of glucose, amino acids, and fatty acids. We understand the architecture. We understand the machinery that breaks the bonds.
We know the what, the how, and the why of digestion.
But we constantly talk about food giving us energy. Somebody burns calories. We obsess over the numeric value printed on the side of a cereal box. But I want you to really think about this biologically.
Yeah.
What actually is that energy?
Yeah, what is it?
Once the glucose molecule enters your cell, how does it become a thought? How does it become the physical contraction of a muscle? It's not a tiny fire burning in your stomach. It's not electricity flowing from a wall outlet.
It is something far more profound, governed by the laws of quantum chemistry and thermodynamics, deep inside the mitochondria.
So next time you sit down to eat, next time you look at a nutrition label and see a number, ask yourself this. What is a biological calorie really? Once it leaves the plate, gets depolymerized into monomers, crosses the cellular threshold, and vanishes into the dark, churning depths of the factory. How does matter actually transform into motion?
That is the true, beautiful mystery of human metabolism.
And that is a deep dive for another day.